To the naked eye, a plant looks entirely still, but zoom in on a growing shoot tip and you'll find an engine of constant creation. Biologists call this active zone meristematic tissue. It is the plant equivalent of animal stem cells, serving as the primary engine for all biological growth. These cells are entirely blank slates. Under a microscope, they appear undifferentiated and packed tightly with dense cytoplasm. They spend their early lives doing exactly one thing: dividing. Every single leaf, branch, and root that makes up the massive structure of a mature plant begins right here. These simple, undifferentiated cells hold the master architectural blueprints needed to construct a towering, complex, mature organism. Eventually, these newly minted meristem cells have to get jobs. They go through a process called differentiation, graduating into specialized permanent tissues with fixed, lifelong functions. Some become simple tissues. Parenchyma cells take on the role of metabolism and food storage, while collenchyma and sclerenchyma cells build thick, rigid walls to give the plant mechanical support. Others join more complex systems. Some stack end-to-end and die, leaving behind hollow, reinforced tubes called xylem. This creates the primary plumbing network, engineered to pull water and dissolved minerals up from the soil. Running right alongside them is the phloem. These are living sieve tubes, responsible for translocating the sugary organic food created during photosynthesis to wherever the plant needs energy. Once this cellular workforce is assigned, the developing plant must commit to one of two completely different architectural paths: becoming a monocot or becoming a dicot. The choice between monocot and dicot is determined before the plant even sprouts, based on the number of cotyledons, or seed leaves, packed inside the seed. That single physical trait dictates the plant's entire anatomy, starting underground. Dicots drive a thick, singular taproot straight down into the earth. This provides a deep, sturdy vertical foundation. If we slice that dicot root open and look at this microscopic cross-section, the vascular tissue is concentrated dead center. You can clearly see the xylem stained green-blue, arranged in a distinct star or cross shape at the core. Monocots take a different approach. Instead of one main anchor, they grow a fibrous, adventitious root system, a shallow, spreading network of thin roots. These foundational differences establish exactly how the plant will anchor itself against the wind and extract resources from the surrounding soil. Moving up from the roots, the plant has to organize its internal plumbing to support vertical height, and here the differences become even more stark. Looking at this cross-section of a dicot stem, the vascular bundles, which contain both the xylem and phloem, are arranged in a continuous, perfect ring around the perimeter. But look at the monocot stem on the right. Instead of a neat outer ring, its vascular bundles are scattered randomly throughout the interior ground tissue, resembling a polka dot pattern. Zooming in on this image of a single monocot vascular bundle, you can see the xylem and phloem packed tightly together. Notice what's missing. There is no layer of meristematic cambium separating them. Biologists call this a closed bundle. Without that dividing layer of cambium, these closed bundles cannot produce new cells for lateral expansion. This scattered arrangement limits the monocot's maximum girth, locking it into a life as a flexible, non-woody plant. Eventually, those vascular pipelines reach the top of the stem and fan out into the leaves, delivering water to power the plant's photosynthetic machinery. This microscopic view of a leaf cross-section shows exactly how that plumbing terminates. That large, dense structure in the center is the midrib, a major vascular bundle that branches out to feed the surrounding leaf tissue. In dicots, those anes branch out into a complex, net-like pattern called reticulate venation. Their leaves also have distinct upper and lower surfaces, with stomatopores concentrated on the bottom to prevent excess water loss. Monocot leaves, like blades of grass, run their veins in parallel lines from base to tip. They also contain specialized void spaces called boulliform cells, which force the leaf to curl inward when water is scarce. At this point, the primary upward growth phase is complete, but for the dicot, a second incredibly powerful active growth is just about to begin. Because monocots lack that vital cambium layer, their stems remain relatively thin, but dicots possess the biological machinery for secondary growth, the ability to expand outward. The key to this expansion is the vascular cambium, a ring of active meristematic tissue. Once triggered, it starts rapidly producing secondary xylem toward the inside of the stem and secondary phloem toward the outside. Look at stages C and D in this diagram. The cambial ring generates new secondary xylem, or wood, pushing outward and crushing older tissues. Annually, it produces large spring cells and narrow summer cells, creating distinct annual rings. To protect this rapidly expanding core from splitting open, a separate layer called the phellogen generates an outer shield of tough cork. This is the periderm, better known as bark. This continuous cycle of secondary growth is the prerequisite for the existence of massive woody trees. When we step out of the microscopic world and look at a forest, the implications of these cellular mechanisms become obvious. Picture a massive ancient oak tree standing next to a field of waving grass. The oak is a dicot built on a taproot, expanding year after year into solid wood. The grass is a monocot, anchored by fibrous roots, remaining thin and flexible. These monumental differences in scale and strength are the direct result of invisible architectural decisions made by tiny meristematic stem cells. Understanding plant anatomy reveals the hidden engineering that allows life to thrive in two completely different structural forms, shaping almost every terrestrial ecosystem on Earth.